Hello, my children. I, Professor Science Person, consider me your guide to the wonderful world of biology. From the inner workings of your brain to the complexities in your cell, this course will delve into the nature of it all. In order to do this, however, we need a base. Therefore, we'll be spending the first few sessions together going over some basic cell biology. So, without further ado, let's jump into the core behind life itself, the genetic code.
Huh, just as I anticipated. Listen, whatever you are. My brain was built to handle the secrets of the stars, so do not regard me with the common peasantry.
Okay, well, don't worry, I'm sure you're very talented. However, it's important we are all on the same page regarding the basic building blocks of life before we jump into the more complicated stuff, don't you think?
Very well, but know that you are dealing with forces far beyond your comprehension.
Uh, Fair enough. Who are you anyway, if you mind me asking?
I, Bartholomew, and I am an Omicron type male, the rarest kind since I made it up for myself.
Congratulations.
Indeed, from your perspective, I am nothing short of a god.
Right, this is going to be a messed up class. Okay, so Bartholomew, what can you tell us about the genetic code?
Well, it is a code used by beings of mortal flesh, and therefore leagues below Miku-chan, the waifu I made on JavaScript.
Whoa. Slow down there. I don't think you're giving the genetic code enough credit. Let's take a quick look. Here you can see the nucleotide, which is the basic building block of DNA. As you can see, it is quite complex. The blue part here is the sugar phosphate backbone. This gives DNA some interesting chemical properties, but from our purposes today, we can think of it as just a structural base. However, the real star of the show is the molecule shown in pink. This is a nitrogenous base, specifically thymine. However, nucleotides can have four different kinds of nitrogenous bases, which are thymine, cytosine, adenine, and of course, guanine. Now, these various bases can then link up via the sugar phosphate backbone, forming a bigger strand, you can see here. Now, the sequence of adenines, thymines, guanines, and cytosines within this strand will then serve to store the information of your DNA. So effectively, the sequence of adenines, thymines, cytosines, and guanines will act as the code within which the DNA can store information.
Excuse me, can I ask a question, please?
Sure thing. Would you mind introducing yourself to the class first, though?
Of course. Hi, everyone. My name is Caleb, and isn't DNA normally represented by a double helix? So why is that only a single one?
What an excellent question, Caleb. Yes, it is true that the DNA is typically depicted as a double helix. Let's see why. So here you can see a DNA molecule. However, in all organisms, DNA is not stored as a single strand, but rather two strands that are bonded together. This is because nitrogenous bases are able to hydrogen bond to other nitrogenous bases, working as a sort of glue to bond two strands of DNA together to form the characteristic double helix associated with DNA.
Okay. Sorry to bother you again, but why would the cell do that? Can't it just store information on a single strand? I don't know if it's a stupid question. I'm sorry.
I no need to apologize. You bring a very valid point. But before I can address it, we need to take a quick look at the DNA molecule once again. As you can see here, the nitrogenous bases within the DNA molecule are forming rungs, like rungs of a ladder. Now, for the DNA molecule to have this nice double helical shape, these rungs need to be of approximately the same size. But as we saw before, nitrogenous bases come in different shapes and sizes. So if any nitrogenous base was hydrogen bonding to any other nitrogenous base willy-nilly, the DNA molecule would form a weird shape that isn't the double helix we all know and love. So there is a rule, which is that adenine always hydrogen bonds to thymine and cytosine always hydrogen bonds to guanine. These two pairs have approximately the same size, allowing DNA to maintain this nice typical shape, even across the millions of base pairs or nucleotides that make up the DNA within your cells. This brings us to Caleb's question, which is what's the point of having two strands? Well, as you can see, we can infer what is on one strand based on what is on the other. So if we have an adenine on one strand, we know there's a thymine on the other. Same with cytosine and guanine, or vice versa. So if we were to split a DNA molecule in half, like seen here, we can predict what is on the other strand based on what is on the first strand. This is how cell replication works.
See, just like that time I correctly predicted the stock market.
Not gonna pretend like you meant that sincerely, but anywho, since we can figure out what is on one strand based on what is on the other quite easily, the cell machinery can do the same. In fact, it can split the DNA molecule in half and then recreate the other half based on the rule that A always pairs with T and C always pairs with G. Therefore, the double-stranded nature of DNA allows it to be easily replicated outside of just storing information. This is what allows cells to produce so many new copies of the genetic material to create huge multicellular organisms such as yourself.
Whoa, so cool. Oh, sorry, I forgot my mic was muted.
Oh, okay, it's heartwarming to see you so excited. So now that we've covered the functions of DNA, how it stores information and how it can be replicated, it's important we cover some other interesting properties. So let's return back to our old friend thymine. Can you see that it is very clearly an asymmetric molecule?
Of course, I can see not only this reality, but through it and into the 9 realms itself.
Can anyone else see that it's an asymmetric molecule?
Yes, but could you explain it a bit more, please?
Of course. So if we take a look at the thymine molecule, we can see that the top and bottom are not equivalent, meaning if I were to flip the molecule around like this, we can see that it doesn't look the same anymore. Now, specifically, we're going to be focusing on the sugar phosphate backbone. Here at the bottom, you can see what is known as a hydroxyl group, which is an oxygen atom bonded to a hydrogen, shown in red. Whilst on the top, you have a phosphate group, shown in yellow. Now, these two groups are important because if we add another nucleotide, as you can see here, we will have one ending that is a hydroxyl group and one ending that is a phosphate group. This relationship does not change regardless of how many nucleotides we add. So if I were to add a bunch more nucleotides, like you can see here, you can see there still is one ending that is a hydroxyl group and one ending that is a phosphate group. Ta-da! But watch what happens when we add the other DNA strand. We can see that the endings are not aligned to each other, meaning on one side, one of the strands will end in a hydroxyl group, which would be this one, and the other will end in a phosphate group, which would be this one. But on the other strand, these are flipped. This means that the DNA molecules are not aligned perfectly to each other, but rather they are aligned opposite to one another, meaning that the DNA strands are anti-parallel. Now, this is not really important to you guys right now, but it's an important concept when we talk about DNA replication later on. So hopefully you keep that in mind.
Excuse me, can I ask something?
Sure thing. The floor is yours.
Excellent. You know, we have been talking about how DNA stores information, but like, information for what? How do we go from a sequence of A, T, C's, and G's to the characteristics that make us us?
Ooh, interesting. Unfortunately, our knowledge is not vast enough to answer this question in full, but we definitely do understand the most important parts. So let's get into it. Although, before we do that, though, what is your name?
I'm a gamer, so I evolved beyond the need for games. But if you want to call me by my gamer tag, then it's Death Reaper 666. Respect. Maybe we could play some Yu-Gi-Oh on top of a moat of lava someday. Oh, or we could do it in my basement, assuming my mom isn't too busy.
Hey guys, let's get back to the topic at hand. So there are many complexities when dealing with the nuclear genome and what it does. But at its core, one of the most crucial functions of DNA is to store information on how to build proteins.
And why are proteins so important?
Well, proteins are the most important part of life itself. You see, they function as the cell's molecular machines able to carry out specific reactions at specific times according to the needs of the cell. These proteins are essentially strings of what are known as amino acids, chemicals that have this composition shown here. Now, we don't really need to get into it too much today, but the most important part to note is the side chain labeled as R. This side chain is different in different amino acids and imparts different chemical or structural properties to the amino acid depending on what exactly it is. Now, depending on the different combination of amino acids making up the protein, it will obviously have different chemical properties. But these amino acids also tend to interact with each other, folding in on themselves to form quite complex shapes. In order to take a look at this, let's look at the protein that I'm sure you've all heard of, hemoglobin. As you can see, the structure is very complex, and apart from looking hella flat, it allows it to carry out its very specific function of carrying oxygen around the body. However, in order to make this very specific shape, you need to know the sequence of the amino acids. So we need some place to store the information of how to make this complex molecule, which is where DNA comes in. You see, DNA is made up of genes. Each gene contains the information to make a specific protein, such as hemoglobin. Now, the A's, T's, C's, and G's stores the information for the amino acid sequence you need to make this very specific structure. But I'm sure you've noticed that, like I mentioned, there are 20 different amino acids, yet there's only 4 bases. So we can't just have every base coding for an amino acid. Instead, bases code for amino acid in triplicate. Essentially, they function as codons, where different combinations of 3 bases after each other specify a single amino acid. So the ratio is 3 bases to 1 amino acid, or 1 codon to 1 amino acid.
However, I have noticed a crucial error. How can one codon code for 1 amino acid when there are 20 different amino acids? But according to your explanation, there are 64 different possible codons based on the A, T, C's, and G's.
Simple, a phenomenon known as degeneracy. Not that kind. You see, a codon will always code for the same amino acid. However, an amino acid might be coded for by multiple different codons so that more than one different codon can code for the same amino acid. This is what degeneracy means in this context.
Okay, but let's say you have a string of amino acids produced from the DNA sequence. How can this sequence assemble into such complex shapes, such as hemoglobin?
I wish I could answer that, but we will be exploring this exact question next time. So stay tuned, everyone, and have a wonderful day.